part of the analysis will be for you to um, learn more information about your particular fish. Now, before we do go on, we're going to leave Subway. So what I'm going to do is I suggest you make your um, um, project public, because if you need help with me or you need to share it with your uh, group mates, what you can do is you go back to your projects and you can send this ID to me or to your group members, and they can search for the ID in public projects and we'll be able to view it. So if you need help or you get stuck, just send me your ID and I'll take a look at it and give you some help. Now, so what I want to do is there's two websites to look at. So one I've told you about is called Flybase, or sorry, not Flybase, Fishbase. And Fishbase is where you can look up something. So I think this is possibly the most closely related of those. So I'm going to go here and search. Okay, and so Fishbase is a international database, so you can learn about the fish that are worldwide and names that they're called around the world. Okay, and you can find out what they're called around the world, what synonyms they have. So this fish was named this by Linnaeus in 1758, but it has been given different names throughout the time until they realized that they were actually the same fish, and so they get lumped under this particular species. You can also, there are a lot of images of the fish, so this is what tilapia looks like, and many different types have been caught. The specimen charts that would be in a museum. Okay, oh, didn't mean to do that. Well, but that's Fishbase, and you can learn about the internationally. Now, if you want to learn more about it, um, about the U.S., what is allowed to be labeled and called in the U.S. if it's sold as a fish, you can go to the FDA. Oh, not that. We go to the FDA, and they have what is called the seafood list, and this is the what fish are allowed to be called, the common names in the United States markets. Let me go back. Okay, we'll give this, takes a little bit of, there we go. Okay, so here we go. Again, the genus and species, here's what is allowed to be called in the United States, only tilapia. It's not allowed to be called anything else. And you can also find some DNA sequence from this, but unfortunately, it's not the same gene. We used the 16S gene. This one is, both of these are the CO1 gene. And so we can't use this in our analysis. So you can see here that it's the CO1, which some people prefer that for barcoding fish. We find that 16S is more reliable, so we use that. Okay, so that's where you learn more information about the fish and what is allowed to be called in the United States. You can also find there's many more websites out there that have barcoding information, and you're more than welcome to pull information from any of those sites. Just make sure that you keep track of where you get the information so that you can put references on your slides. Okay, so that's how you can analyze the data, find out what your fish is, and see if it was labeled correctly. Remember, you want to go back to the spreadsheet and see how it was labeled. Then look at the relationships amongst the different fish that we have in the class, And if you want, you can look at all the fish sequence that you have been assigned. You can make the project as big as you want. That would be cool. Lastly, what I would like the group to do, each group, is do some research online about how DNA barcoding is used in other ways. So identifying fish species is not the only way. It's used in medicine for all kinds of interesting applications when you can't necessarily clone genes or isolate organisms. It's been used to determine if the meat in sausages or what's actually on the label. It's been used for all kinds of basically regulatory, but also medical and environmental and studies. So go out there, see what you can find about how barcoding is used throughout the different disciplines, and give us a little case study, just one slide on what they used it for and how what they found. Okay, I'll be meeting with the groups and I'll be providing more detailed information on the presentations in the next couple of weeks. But I'll definitely try to meet with all the groups next week and check up and see how the sequence analysis is going along. Okay, talk to you then. Bye.